All right, hello everybody, and welcome to the next topic, which is 2.1.2 within the molecules to metabolism section. We're talking about 2.1 here. We're going within 2.1 molecules to metabolism. We are now breaking it down even further, and we are going to talk about one of the biomolecules, which is known as carbohydrates, that I'm sure you have heard about. We are just going to go through the absolute basics of carbohydrates. We will go into it a little bit more when we get into its very own topic later on in this series of videos. Okay, so first things off, we've got these words that I want to deal with right off the bat. One of the words is called a monosaccharide. Now, let's just break down this word. The word mono obviously means one. The word saccharide means sugar. So this is a sugar that has one ring of carbons, and I'm going to explain that in a second. Some examples of monosaccharides. You need to know these examples. An example of a monosaccharide would be something like glucose. Another example of a monosaccharide would be something like fructose. Another example of a monosaccharide, galactose. These are classic examples of monosaccharides. Now I'm going to give you a very basic demonstration. Let's say glucose looked like that. A little red dot. Fructose looked like that, and galactose looked like this. A basic understanding. If I take one glucose molecule, okay, and I go and find another glucose molecule, there it is over there, I can join these two glucose molecules together with a bond. So join them up with a bond. This is now no longer a monosaccharide, meaning it's not just one sugar, it's two sugars. When it's called two sugars, it's called a disaccharide. Glucose and glucose. This particular disaccharide is called maltose. It's when you have two glucose molecules together. Let's take another example of a disaccharide. Let's take glucose. Don't know why I copied that. I'm just going to draw it. Let's take glucose and let's take fructose now and put these together. Join it up with a bond. This bond you need to know, the bond that joins two monosaccharides is called a glycosidic bond. And now we've got a different disaccharide. This disaccharide, when you've got glucose and fructose together, is called sucrose. And this is the kind of sugar that you find, that you put into your coffee and your tea that you find in most households. You do get something called a polysaccharide. See? Polysaccharide. Let's do this somewhere else. Let's do disaccharide examples. We've got sucrose. We had that one earlier, maltose. You get lactose as well. Lactose is made up of a galactose and a glucose molecule. I'm not going to draw that. You can draw that yourself. That's a disaccharide, two sugars. We're going to go into exactly what these look like in terms of the chemical formulas, in terms of what the ring looks like, and I'm going to tell you exactly what you need to learn for your IB exam. Those are disaccharide examples. Let's do some polysaccharides. So polysaccharides, of course, would be these long chains of monosaccharides. So you could get, I don't know, maybe 5, maybe 6 glucose molecules. 1, 2, 3, 4, 5. Doesn't really matter how many you have. The idea is that you've got more than 2, meaning it's a polysaccharide. So they're the glycosidic bonds. I'm drawing really badly. This is an example when you've got a lot of glucose molecules together. Examples of these are things like starch, glycogen, not glucose, but glycogen. Very common mistake for students to make, to get that word mixed up. Another example that you find in cell walls of plants is cellulose. You need to know these examples of polysaccharides. Beautiful. I hope you're understanding that monosaccharides just have one of these sugars, disaccharides have two, and polysaccharides have many or more than two. Okay, so let's go into the basics of a few of these molecules. Let's take, for example, a glucose. Okay. The basic structure of glucose. This is how I do it. I know that it has the formula C6H12O6, one of those nasty things that you're just going to have to go and remember. It's not too bad. This is how you draw glucose. You need to know how to draw an alpha glucose. I'm going to explain what this is in a second. You need to know how to draw an alpha glucose molecule. You need to know how to draw a beta glucose molecule, or beta, some people say beta, and then you need to know how to draw a ribose. This is another example of a monosaccharide that I left out. Let me go and add it. Ribose. Just another example. Let's get the colors going. Maybe yellow. Beautiful. Okay. You need to know how to draw these structures. Let's go and do this. Okay, first off, it contains a ring. So it's basically like taking a whole bunch of carbon atoms that were all joined together, but now you're kind of bending them around and you're joining it up into a nice ring. Let me show you how to do it. First off, draw yourself a nice hexagon. So you got one line here, joining it up like a nice good old hexagon, except that this line is going to go to an oxygen. Now, what do these little joins mean? In chemistry, we can sort of take a shortcut, and instead of drawing carbon bonded to carbon, bonded to carbon, and you're welcome to draw it like this, bonded to carbon, bonded to oxygen, bonded to that carbon, you can sort of draw a more condensed version by just drawing the lines. You don't put these little dots, you simply just draw the lines. Let me draw them again. Okay, so go down, down, down. That's not the right way, that's a different structure. Down, down, across. This one goes to an oxygen, that one goes down to there, and that one goes down over there. You've drawn a ring of carbons, and this carbon is joined up to that oxygen, and this carbon is joined to that oxygen over there. Excuse me. Oxygen, we know carbon has a total of 4 possible bonds. Oxygen's got 2, and there are many other elements that have different amounts of bonds as well. I will do a chemistry video on all the different elements and how many bonds they can have, and the reasons why carbon has 4 and the reasons why oxygen has 2. For now, I would not worry about it. Okay, so I think let's work with this structure at the bottom over here because it makes a lot of sense. We now have 4 possible bonds at this carbon. One there, one there, one there, and one there. Oxygen is happy. It's got its 2 bonds, so nothing more can possibly join here. Over here, we've got another 2 possible joins. Over here, 2 more joins. Over here, 2 more joins. Can you see it's going to get a little bit messy because now we've got to write an element over here and an element over here as well, so it's going to get a little bit messy. So try to draw your rings quite big, and then we've got the same thing happening over here and the same thing happening over here. So just right from the start, let's do it all again. Do it together. We've got a carbon bonded to an oxygen, which is bonded to a carbon, 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 and then this one joins up with that carbon. Okay, great. Now, we have a varying amount of carbons here. We've got 1, 2, 3, 4, 5, and this carbon on the right is called carbon number 1, or carbon 1. This is carbon 2, 3, 4, 5. But I know that my formula for glucose is carbon or C6H12O6. So we have to include another carbon. The other carbon is joined to this carbon 5 over here, and it's joined in such a way like that. That's carbon 6. This, of course, has 4 bonds as well. 1, 2, 3, and 4. Okay? So right now, we've got a whole bunch of places that we need to fill in. alpha glucose, the defining feature of alpha glucose is that it's got this hydroxyl group. When you've got an O and an H bonded together, it's always O first, then H, not HO. This is called a hydroxyl group. Lots of stuff to learn, and I'll give you an explanation in the chemistry video of why it's called a hydroxyl group. In an alpha glucose, the hydroxyl group, the OH that's going to be bonded to carbon 1, is bonded here at the bottom. You draw an O, carbon's bonded to that O, and this O is bonded to an H. In some textbooks and in some videos, they don't even show this bond. They just simply put the H over here. It doesn't mean that it doesn't have a bond. It does have a bond. It's bonded to, I need to drink some water. Excuse me. It's bonded to a hydrogen over here. This carbon here is also bonded to a hydrogen. So this carbon is happy. And carbon 2, you have another OH bond. You need to draw this OH bond at the bottom over here. On this carbon 2, there's a hydrogen. Then on this carbon 3, the OH group that's on this carbon 3 is above within this ring over here. And then a hydrogen over here. And then this side on this carbon 4, you've got the OH that's bonded over here. Now I can go OH like that, showing my line is joining to the oxygen, and then I can draw a little line bonded here, or I can draw it like that. I'm going to show you the condensed formula. Excuse me. I can show you the condensed formula in a second. And then we've got a hydrogen over here. This carbon has it's bonded to an O, which is bonded to an H, and then there's two H's over here. That's how you draw a alpha glucose. Now, I know this looks kind of messy, so let's try. Oh, wait, we've forgotten about this carbon over here. This carbon over here has a hydrogen, and there's too many bonds here. Oh, there's no bond there because you've got 1, 2, 3, 4. Okay, now I'm going to start to condense things. I'm going to take these OH, this O that's bonded to this H. I'm going to remove that and condense it so that it looks a little bit nicer, but still knowing that that oxygen is bonded to a hydrogen atom. You just don't have to draw that line. You can do that for all the OHs. Okay, it's always OH. I'm not reading this HO. I'm reading from right to left. Again, over here we've got the OH, and these OHs inside. That's an OH there. This whole group, you can actually condense this whole thing into C, being this C here, then the H2, these two H's here, you can say H2, and then the OH. So if you condense it, it's like that. If you break it up, it looks like that. But I'm going to condense it. And in your drawings, in your IB exam, you can condense this section as C. Make sure it's joined to the C, not to the H. CH2OH. That's the 6th carbon over there. That's what gives us C6. Okay. This is alpha glucose, making sure that the defining feature of a alpha glucose molecule is that this OH, I don't know why I've highlighted the carbon, this OH is below this carbon, okay? It's going downwards. In a beta glucose molecule, it's switched around. So you would draw the OH over here and the hydrogen over here. I'm going to do the beta glucose in a second. Okay, so this is the structure of an alpha glucose. I'm going to start from the beginning, and I'm going to show you how simple it is. Once you start doing it a couple of times, it becomes really easy. Okay, I'm not going to draw C's, I'm just going to draw lines. So I've got that first one. There's my carbon 1. This carbon is bonded to an hydroxyl group. It's going below. It's also bonded to a hydrogen. This then joins up over here, joins up over there, joins up over there. I've got the CH2OH group coming off there. I've got my hydroxyl group coming off here. I've got another hydrogen going there. I know that there's an OH group over here. There's an OH group over here. And there's a hydrogen coming off here. There's a hydrogen coming off there. And I think I've got myself a complete alpha glucose molecule. Let me just draw a line down here so I can draw my beta glucose. Now let's just go and count up all the carbons. 1, 2, 3, 4, 5, being this point over here, and 6. So I've got C6. Let's see if we've got the H12. I've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. I'm missing one here. 11. I must be missing another one somewhere. Let me see. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. I've got my H12. The oxygens, 1 over there, 2, 3, 4, 5, 6. I've got my perfect molecule. So that's where they get this breakdown from. Now let's have a look at beta glucose. Everything exactly the same, except you're going to switch these two around. So we've got this carbon 1 over here, and it's specifically carbon 1 that you switch the hydroxyl group. The hydroxyl groups on everything else have to be in those positions, and they stay the same. Now my OH group is going up here, my hydrogen's going down here. Can you notice that it's switched around? That's when it's beta glucose. Okay, and draw like that, like that. Then my hydroxyl group's still the same. OH over there, OH over there. I've actually seen in tests and exams that they've left out the hydrogens. And you've just got to observe this hydroxyl group here, and you've got to say, okay, that's beta glucose. Oh, that's alpha glucose. Just literally looking at that carbon 1, making sure that these hydroxyl groups are also in the right place, but simply looking at this carbon 1, you're able to identify whether it's alpha glucose or beta glucose. Of course, you've got to have everything else in place as well. So that CH2OH group over there, you've got a hydrogen over there, you've got a hydroxyl group coming off here. Of course, we've got a hydrogen there. I'm going to include my hydrogens in this diagram. Hydrogen over there, and I think we've got ourselves a complete beta glucose molecule. You need to know how to draw this. You also need to know how to recognize it. A ribose looks completely different. It's a pentose. These both are called hexose molecules. Hexose, because hex meaning 6. It's got 6 carbons. A ribose only has 5 carbons, so it's called a pentose sugar. Okay? Sugar meaning it's carbohydrate. So we start off drawing a pentose. I like to think of it like a little house, and it's like the structure, like a pentagon. Okay, so they got 1 carbon there, 1 carbon there, 1 carbon there, 1 carbon there. That's 4 carbons. We need another carbon. And this is called ribose. It's in the category of a pentose sugar. So you get a whole bunch of pentose sugars. The only one that you need to know how to draw is ribose. Okay, we've got the CH2 OH group coming off this 4th carbon over here. We've got a hydrogen coming off there, of course, because it's a carbon molecule. It has to have 4 bonds. Again, we've got these OHs coming off the bottom that look like this. You need to know that. You need to know that the OH groups are coming both off the bottom. This one, the OH group is off the top. And there you have your ribose structure. Go and learn these 3 structures. Pretty sure that I've got this correct with all my hydrogens. And this would have the formula C5H10O5. Notice that hydrogen to oxygen ratio, 2 to 1. 2 to 1. The beta glucose is also going to have the same formula. Okay, 2 to 1 with the hydrogen to oxygen ratios. Notice that these have the same chemical formula, but structurally they look different. We call things that have the same chemical formula but have structural differences, these are called isomers of each other. Same chemical formula, different way in which the atoms are arranged. Okay, great. So let's move on. Let's carry on talking about some of the other characteristics of glucose. So this is kind of just like if you wanted to summarize glucose, remember it's a monosaccharide. So let's get that as a heading. So monosaccharides or the monosaccharides. Glucose is the primary source of energy for living organisms or living cells. Primary source of energy for living cells. So if you had a disaccharide, I don't know, let's say like we got maltose here. Sorry, I've just drawn that glycosidic bond with a red pen. Just ignore that. If you ate a disaccharide, so you ate some maltose, your body will break down. It will chop up this bond effectively, and so that you've got this primary source of glucose, so that this glucose molecule can enter your cell and be metabolized, and it can get used for energy. So glucose is a primary source of energy for living cells. It's a monosaccharide, and it also forms under the umbrella of hexose sugars. Okay, 6 carbon sugars. Let's go and look at fructose. So we've got fructose again under the category of monosaccharides. It's also a hexose sugar. You don't need to go and learn the diagram of fructose. Again, just glucose, the alpha and beta versions, and ribose. Fructose, the only thing you need to know is it's a monosaccharide. It's the sweetest sugar. It tastes a bit like honey. So sweetest sugar, tastes like honey, and that's about it for fructose. We've got ribose, of course, that's another type of monosaccharide, except that it is a pentose. It's got 5 carbons. And we are going to learn a lot about genetics later on, and RNA is made using ribose. That's just a fact. Remember, we are only skimming over the basics of carbohydrates, so this is just an introduction to carbohydrates so that you've got a little bit of the basics before we go into the depths. Let's go and look at the disaccharide sucrose quickly. Disaccharide. Sorry, I'm getting more and more unneat as this presentation continues. Disaccharides, let's look at sucrose. Remember, sucrose is made of glucose and fructose. So it's when these two things combine together, you get sucrose. Glucose and fructose. And this is a sugar. It's combined with it's got a glycosidic bond, of course. Remember, there's the glucose, there's the fructose. And in between these two monosaccharides, you have this glycosidic bond. We're going to go into how this actually works in terms of the ring structures. We're going to go into these types of reactions later on. And plants actually use sucrose in terms of how they transport sugar. So plants transport, when they want some sugar to go to the top of the leaf, for example, plants will transport sugar as sucrose. So this is just some facts. Plants transport sugar as sucrose, and it's combined glucose and fructose together. Great. Let's go on to the polysaccharides. Saccharides, we talked about these earlier, but let's do some examples again. We've got cellulose. Cellulose kind of looks like this. It's like a whole bunch of these monosaccharides, whole bunch of these glucose molecules just joined up together in this long chain. And you actually use cellulose in plant cell walls. So you'll find cellulose in plant cell walls. It's used to make plant cell walls. Of course, we've got glycosidic bonds joining up all of these monosaccharides. Some terms sometimes get used that this is a monomer, and the whole thing, I never know how to do one of those squiggly lines like that. The whole thing is called a polymer. Okay, and you can use these words across the board when you're talking about various different domains. What I mean by that, you can use it in other subjects. You can use the word monomer and polymer. It doesn't necessarily mean you're talking about polysaccharides, but it can be used for that as well. We've got another example of a polysaccharide, starch. You would have heard of starch. Plants store polysaccharides as starch. Starch looks a little bit more interesting. We call this, when it's in one long chain and it doesn't have any branches coming off, like think of it as like a long tree that has no branches. We call this unbranched. So cellulose is unbranched. That kind of makes sense. If it's making cell walls that are nice and long and straight, we want a molecule that is nice and long and straight. So we've got cellulose. Starch, a little bit more complicated. It's got a whole bunch of these monomers in a branched fashion. So things branch off from each other. Let's go and draw those glycosidic bonds. I don't know why I've made the spaces different. Should all be the same spaces. Can you see the branches coming off now? So we say that this is branched. Okay, and we know that plants store sugar, or you can say plants store carbohydrates slash, I'm just going to say carbs. Don't write carbs in your exam. Write carbohydrates as starch. Examples of starch, we know potatoes. So potato, we've got things like corn, rice, bananas. All these things are examples of starch. Beautiful. And then the last thing I want to talk about is when you combine a protein plus a polysaccharide. This can happen. It's called a glyco. That's the polysaccharide version. Glycoprotein. So it's what is a glycoprotein? It's a part protein, part polysaccharide. Just something to mention so that when it comes up later, it's not a big shock. Beautiful. Let's have a look if I've left anything out of my notes. That's pretty much everything I've covered today. You should have learned how to draw the various different diagrams of glucose and ribose. You only need to know the ring forms of ribose, alpha D glucose and beta D glucose. Don't worry about the word D. It's just something that you might see. It's just alpha glucose and beta glucose. The D is just something to do with the chemistry in terms of how the atoms are arranged. Let's not worry about that for now. Wonderful. I hope it was helpful, and on to the next section, we're going to be talking a little bit about proteins.